9.5, Paul Boutros.
Okay, so I'm going to announce two ICGC-TCGA Dream Somatic Mutation Calling SMC challenges, both to start in January, February of 2015. The first is the SMC RNA challenge. It's open for registration now. And the process of this challenge will involve data distribution and some cloud-based aspects, exactly as the SMC DNA challenge that I talked about this morning did. It will resolve, or will resolve, I hope. It will address the biggest issue that we got questions about while we were running SMC DNA, which was, does all of this count for RNA? Is RNA the same? Can you do the same thing so that we can standardize methods? And so we're going to be looking at three key questions. The first, whether we can detect the presence of fusion proteins, especially cancer-specific fusion proteins, since that's the vast majority, from RNA-Seq data. Can we do isoform reconstruction in tumor RNA-Seq data? And lastly, and a little bit less, more of a side challenge is, can we identify somatic single nucleotide variants from RNA-Seq data? People do this for a number of reasons, one of which is to validate genome sequencing data, another one of which is to look at RNA editing, and it turns out to be a nice addition to the first two. This will all be RNA-Seq data. There will be 100 tumors derived from 4 separate tumor types. Although 100 sounds like a lot, in terms of raw sequencing data, it's exactly the same size as the SMC DNA challenge, so it's not growing the disk space envelope. There will be a retrospective experimental validation in very much the way that I described this morning for SMC DNA, and that will be used to generate a gold standard for the community, as well as having a scoring of the algorithms. The timeline will be the launch today for people to begin registering. The competition will run from January through to July of next year. We anticipate just a 1-month experimental validation, although it might end up taking something like 6 or 7 weeks, and then announcing a winner in September or so for challenge presentations at this meeting next year. The challenging organizing team includes all of those people who worked on SMC DNA, myself, Josh Stewart, Adam Margolin, Gustavo and Stephen from IBM and Sage, and importantly also Matt Wilkerson from UNC, who is an RNA-Seq expert who will be working with us on this. And we have many funding partners and data contributors who are participating to help this challenge get launched. Similarly, I'm also excited to announce the other challenge that we're launching at the same time, which is a tumor heterogeneity and evolution, what you might call a subclonal reconstruction. This is the third thing that we kept on getting questions about while SMC DNA was going was, you should look at subclonality in more detail. And so this challenge specifically addresses that. It also opens for registration today. And it's really interesting because it's a cloud-only challenge. We're making available a Galaxy interface and an API. Groups will be submitting their code directly into the cloud, and all of the running of the code will be done in that interface. This is something that Kyle Elrod and Josh Stewart's group is leading the development of and creating a pilot for it right now to make sure that we're ready to roll it out in January, February. The challenge has 4 sub-challenges. The first to determine the number of subclones within individual tumors. The second to determine their mutation order, the precedence at which these occur. The third is to estimate the subclonal proportions, how much of each of these different tumor populations there are. And then lastly, and most difficult, to predict the specific evolutionary tree in the lineage that relates all of these together. Like SMC DNA, this will have 2 parts to it. There's an in silico component, which is 50 whole genome sequences. These are all simulated in much the way that we described today. They're again of differing complexities, and they incorporate different parameters about the underlying tumors. So for example, they will have different amounts of subpopulations, they will have different depths of evolutionary trees, different amounts of normal contamination within the sequences, and that'll allow us to try to get a feel for just how different algorithms work on different problems. And because it's an automated challenge, the code will be available so that if we want to start adding additional tumors into the challenge to test specific things, all the code will be there available for ready running of it. Additionally, there will be single-cell sequencing data from 11 data sets. It'll end up being 13. There's another 2 that are in progress. These capture ovarian, leukemia, and breast cancers and allow us to not just be using simulated data where we think we know the truth, but data where experimentally it's been validated. The challenge organizing team includes myself, Quaid Morris from the University of Toronto, Josh and Adam. Kyle Elrod, as I mentioned, is playing a key role in helping us work out the automation aspects of it. And now we'll hear about the Dream Olfaction Prediction Challenge.
Hi, my name is Pablo Meyer, or Paul Meyer, depending on just following 2 Pauls. This is part, I think, of kind of the diversification of Dream in a sense. And so I'm kind of now looking over challenges that we call are related to emerging data. And so while giving a talk about Dream and about other things, at Rockefeller University, somebody approached me and told me, oh, we have this great data set that we're developing about olfaction in humans, trying to map olfaction in humans, but we really don't know anything about computational biology and we don't know what to do with it. The only thing I have is this huge Excel file that I can barely open and I don't know what to do with it. What do you think? You think we could do a challenge? And lo and behold, I mean, little by little we realized that it would be a great challenge. And so here I am now presenting the Dream Olfaction Prediction Challenge. And the idea is that we want to predict the perceived smell of molecules based on their physical and chemical properties. And why do we need this challenge? Well, we need this challenge is basically these 2 molecules, benzaldehyde and hydrogen cyanide, have very different structures, but still smell like almonds to us, both of them. And we don't understand why. So there's a problem of trying to map the chemical space to the perceptual space. And of course, this is a very interesting problem because, we like to smell good things and we like to, and companies would like to try to understand why some things smell better than others for us. So the amazing data set that has been collected by Andreas Keller and Leslie Volsheil's lab at Rockefeller University consists of 49 subjects that were presented, 480 orders at 2 different concentrations, but we're going to separate the set into test and training set. So there will be 240 orders, 2 concentrations for doing the training, and each of the stimulus were presented to the subjects, and they had to rate on a scale from 0 to 100 how strong the odor was, how pleasant, and then other 20 different descriptors that are actually pretty funny to just read. Edible, bakery, sweet, fruity, fish, garlic, spices, cold, sour, burnt, acid, warm, musky, sweaty, ammonia, urinous, decayed, wood, grass, flower, chemical. So these are the 20 descriptors that were presented, and basically they just had to scale and try to define what was the stimulus. And also these 480 orders were actually selected in order to try to cover the other space. So, as the same way as we have a visual space of colors, we are supposed to have a other space that's not very well defined, and even the measures and the size of this other space are not well defined and actually even contentious. But the idea is that we have around 400 GPCR receptors that are, each of them is specific for a single odor, and so the idea is that with these 480 odors, we will cover the whole space. And we will also provide molecular descriptors of these molecules. So the advantage, these are single molecules, no mix of others or anything like that. And so you can clearly define, with chemical angles and distances between the atoms and etc. You can define what these molecules are. And so the organizing team is Andreas Keller that basically put together this study, Leslie, who's the head of the lab, my colleague Guillermo Cecchi at IBM Research, André Falcao at Universidad de Lisboa, and Gustavo's has also been part of the organizing team, always helped by Tia Norman and, well, myself. So thanks a lot. Hope you like it. Yeah, this is part of Dream 10. 9.5? Okay, sorry. No, sorry. It's only half a mistake. So, so now I'm pleased to introduce the next challenge presenter, Lara again. Hi.
All right, so we want to give you lots of diversity, so you've got plenty of challenges to select from. So I'm here to introduce the Prostate Cancer DREAM Challenge, which is also a DREAM 9.5. So this is a collaboration with Project DataSphere, which is an organization which is working to release comparator arm data from clinical trials run by pharmaceutical industry partners. And so they've put together a prostate cancer data set coming from 4 clinical trial data sets coming out of industry. These are coming from Sanofi, Celgene, Memorial Sloan Kettering, and AstraZeneca. So this is metastatic castration-resistant prostate cancer. And the question is whether we can actually predict survival in these patients based on the rich clinical information that comes out of these clinical trials. So I think that this is a direct parallel to what we saw with the rheumatoid arthritis challenge this morning. So in this case, we have clinical variables only, but we have a rich plethora of clinical variables. So on the order of hundreds rather than a half a dozen that we had this morning. So given that we know that clinical variables are very important in prediction, can we just focus on them and see what sort of predictive models we can build out of that? So similar to RA, we're breaking this into 2 questions, predict survival based on clinical variables, and then classify the patients into 3 groups, so good survivors, medium survivors, poor survivors. And then we have a third question, which allows us to take advantage of some of this clinical trial data to actually look at whether we can predict treatment discontinuation for people treated with docetaxel. So as I said, there's 4 prostate cancer data sets. 3 of them are being combined right now in a curation effort, which is no small feat, into 1 training set, and then we'll hold another one back as the validation data set. This is meant to build on the work that's already out there. There's a rich academic history of looking at predictive modeling within this field, and so the best I think the best regarded model that's out there right now is by Susan Halaby from Duke. And so she used these 8 factors to build a prediction of overall survival. So this is going to be our baseline. We are all looking to see as to whether we can incorporate additional information to do better than this. There's a wide range of people involved in this. The leads of this challenge are actually Justin Guinée at SAGE and Liz Zhu, who put this slide together and therefore did not put herself on this slide, but she's at Project DataSphere. And we have a wide range of people who are involved here, and we think it's shaping up to be a pretty exciting challenge. So pre-registration is open today, and we expect that the challenge will launch around February 1st, so stay tuned. Thanks.
All right, so that's more or less all that we have aligned for DREAM 9.5. That's 2 SMC challenges, 1 challenge that doesn't smell very good, and 1 challenge that is on prostate cancer. Now, for DREAM 10, we are already preparing some challenges for DREAM 10 I think are very interesting, some of which are heavy in imaging, but we'll announce it at some other point. What we definitely can announce now that is most likely going to run during the fall of 2015 is the ALS2 challenge, or the ALS Stratification Price for Life challenge, which is a follow-up, a sequel to the previous ALS progression prediction challenge that was just a week ago, published in Nature Biotechnology. So the idea is to leverage the incredible aggregate of clinical trial data that this Foundation Prize for Life has collected, and use that data in order to try to make inroads in the prediction of the progression of the disease, trying to learn how to stratify the patients in intelligent ways. So one of the things that I'm often asked is, how do you get the funding for the challenges? And so there are many ways in which you can think of getting the funding. None is trivial. One that we are thinking for the ALS is to use the Indiegogo crowdfunding website in order to get the interest of people who are, if people throw themselves a bucket of ice in their head with the ice bucket ALS a campaign, then we hope that they also would be interested in putting a little bit of their hard-earned money into trying to fund the ALS stratification challenge. So, and that is it for the DREAM 9.5 and DREAM 10 challenges. Any question about these challenges, when they will start, what the incentives will be? We don't have answers to any, but if you have questions, we would like to entertain them. I don't think so. So, okay, so here is what we are going to do. Please, let's go to the next room where there will be some pizza, probably now or in 5 minutes. I don't know whether it's already there. And then please come back with the pizza and the beer here, and we will start a little discussion as to where we go with DREAM in the next year. Thank you.